Zoom talk of Eric. So actually, Eric could just, if he hears us, he could start sharing content. That's fantastic. I will quickly introduce Eric. He studied marine natural products chemistry at the Scripps Institute with Professor John Faulkner, and then he did his PhD at the Johns Hopkins University with Greg Townsend. And since 2001, he is member of the Department of Medicinal Chemistry and Biological Sciences at the University of Utah, and is currently professor and Droschke endowed chair in pharmacy. So we are now moving further west. So we are now in Salt Lake City. Eric, it's a pleasure to have you here. We have seen very exciting recent work on megasyntheses, so we are all very excited to now listen to your talk. So thanks for being here.
Okay, thanks very much for having me. I'm excited to be here and have been really interested in all of the talks that I could make. I did watch the sun come up this morning during the symposium, so it's difficult to catch the earlier ones. I do miss being there in person greatly. While I was watching these talks, I was reminded of about 10 years ago being at a meeting and sitting next to Adrian on a bus talking about methylated polyketides in animals. And so I miss those rewarding experiences and hope we can be back to those soon. So I'm going to tell you about polyketides in animals. For this audience, I thought it was worthwhile to show you a phylogenetic tree of animals. And we really think about these as a diverse group with millions of species. It's like studying the actinomycetes or the gamma proteobacteria. They're extremely biodiverse. You're starting from simple forms like sponges, going through all kinds of different animals, insects very rich in chemistry, starfish, worms, and all the way up to humans. So there's a lot of biodiversity in the animals. And one thing that we noticed while working on this project is that all of the animals also have their own unique natural products chemistry. A lot of what we know about animal natural products comes from systems where we can obtain the animals and find compounds in abundance. And this is really a relatively small percentage of the animals that are out there. So one example that's near and dear to my heart is shown here, the patellozoles, first isolated by Chris Ireland and co-workers. These are compounds that are quite complex. You can see they're a mix of polyketide and non-ribosomal peptide metabolism. They're found in this tunicate animal, which is a chordate quite closely related to us. Chris found that these are picomolar in their anti-cancer activity, really potent, and they're also found in great abundance. For example, here is an LCMS trace where you can see that the most abundant extractable compounds are these picomolar toxins. So we know a lot about compounds like these from animals. And this is work done by Jason Kwan back when he was in my lab an age ago. And a lot of what we knew at that time and a lot of what we continue to study and other people continue to study are how the complex natural products such as polyketides and others that are found in animals are really made by symbiotic bacteria and not by the animals themselves. And so in this case, Jason cloned the biosynthetic pathway and he looked at a slice of the animals. And what we can see is that the bacteria that make patellozoles are proteobacteria that are packed into white blood cells in the tunicate. And so there's this really specific and long-standing relationship focused on chemistry. Excuse me. But what we have been more interested in lately are the many PKSs that are actually encoded in animal genomes. This is an area that's been quite challenging, but recent technical advances are making it more approachable. So when I think back to what's known about animal PKSs, I think the first one that was really described in any detail was probably this one from sea urchin, described by Christina Calistani and co-workers in 2003. And I think she's really probably the pioneer, or at least a major pioneer in this field, and that she identified this as a PKS and speculated that it might make this compound echinochrome, which is responsible for the coloration in sea urchins. Since that time, there's been, especially recently, a lot of work focused in part on knockout mutagenesis. And so I'm showing you one example where knocking out some of these genes, such as PKS, lead to loss of coloration in the sea urchin. So there's a very compelling but still indirect connection between this PKS and the echinochromes. You can see that these PKSs are iterative and have kind of a fungal-like flavor to them. A famous example is one that was first identified by Maureen Hillenmeyer in 2013, and then further worked on by Rebecca Butcher more recently. And I think Maureen really came a long way in recognizing this PKS NRPS hybrid encoded in the genome of C. elegans. And you can see that there is an iterative and a modular nature to this hybrid PKS NRPS. When we went back and looked at the KS domains, this is clearly an animal-like PKS, but fused to more complex machinery. And then the Butcher group isolated these compounds, the nemamides, that were associated structurally with the presence of these genes. So again, indirect, but very compelling and exciting work. One of my favorite compounds in this area, just because it produces this famous dye, carminic acid, the beetle that produces carminic acid, I should say. In this case, there's no PKS yet identified, at least that I'm aware of. But there was a paper a couple of years ago by Kanengara et al., where this carbon-carbon bond forming enzyme was characterized out of the animal genome. So providing some compelling evidence that this compound is probably also animal genome encoded. So what all of these have in common is that they're fairly iterative in nature in terms of the PKS. And that work continued more recently through genome-wide studies, genome-wide mapping. A PKS responsible for the yellow pigment in some birds was identified, leading to this polyene. And again, an iterative flavor of PKS leading to this polyene. And there are a lot of others in the literature that you can find that have some kind of partial characterization where there's some association with a compound, for example, in fish ear development and in other places. So even vertebrates have these PKSs. We've been working on this for a couple of years, and this is one unpublished thing that we've been working on more recently, a phylogenetic tree made by Joshua Torres and Iris Uy when she was in my lab. And this is an updated version of the animal phylogenetic tree of PKSs. What you can see is that they're everywhere, that there are many families distributed everywhere. And also, there are what appear to be modular PKSs encoded in animal genomes. So this idea that they're bacterial and they're not found in other organisms, I think, is probably not going to stand the test of time. I think part of the reason why these are being uncovered now is that they're quite hard to assemble in the animal genomes quite often. So for example, here's a modular one leading to no known product found in the sperm whale, ending with a sulfotransferase, which is interesting. Here's one from the Virginia quail or northern bobwhite found in the US, and you can see this kind of typical modular structure ending with a reductase domain. So there's a lot of complexity out there that's yet to be uncovered, and that's very rich and interesting, awaiting discovery. So when we were coming into this project, we had a couple of questions about the animal PKSs. I think I showed you in the introduction that there's really nice work that's been done by many groups, but there was still very limited biochemical characterization of these enzymes. We really wanted to be able to express these things and have a close look at them. In addition, we wanted to know if animals can make truly complex polyketides, or whether they're limited to these relatively simple products. In the case of the PKS, NRPS, it's still relatively simple, but fused to a complex polypeptide. Nonetheless, these kind of malonate-derived iterative products are not the same as what we anticipate that bacteria can make. So our first project in this area, and the one that I'll tell you about today, focuses on the tridachiones isolated by Chris Ireland in the 70s. Tridachiones come from mollusks, these sacoglossan mollusks. And what you can see about them is that they do start to have some structural complexity. They're methylated, and they have these pyrone head groups. So Joshua Torres, when he was a graduate student in my lab, spearheaded this project and did most of the work and came up with most of the ideas. He's now a postdoc at the University of Copenhagen. Zhenjian Lin is a research professor who did lots of key experiments in this process. We got a lot of input from assistant professor Jacqueline Winter in our department as to how to express and handle the PKSs, and our slug expert Patrick Krug from Cal State LA. Our work focused on the Sacoglossans. These are solar-powered slugs. What they do is they eat algae and they digest the algal bodies, but the chloroplasts are not digested. Instead, they're diverted from these gut-derived tubes where they can perform photosynthesis for up to months and keep the slugs alive. There are many different flavors of Sacoglossans. The ones that we were most interested in and that we'll talk about here are the long-retaining Sacoglossans. They can maintain the chloroplasts alive for months, and there's experimentally documented significant photosynthesis coming from these slugs. So the slugs are able to live for months on photosynthesis alone. On the other far end, there are the non-retaining slugs. These eat algae but don't retain the plastids. And in the middle, there are the short-retaining organisms. These can retain chloroplasts briefly, but there's little or no documentation of photosynthesis being important in their survival. We liked these groups because there's quite a good pre-genomic days biosynthetic literature, starting back with the work of Ireland and Scheuer in 1979. And they did a couple of very important experiments. One thing they did is they fed these captive slugs with radiolabeled carbonate. And they demonstrated that carbonate was taken up by photosynthesis from the chloroplasts, and that it was incorporated de novo into these polyketides. So they showed that it was de novo. They showed that photosynthesis was important. Another thing that they did is they looked at the photo rearrangement chemistry. And so they showed that if you expose these compounds to UV light, that they undergo rearrangement. So at this time, they had proposed that the role of these molecules in nature might be to photoprotect the animals. These animals need to be exposed to sunlight and shallow water in the tropics. They're soft-bodied and have no shell, and so they're very vulnerable. And so one idea was that these compounds might be photoprotective to this process. There have been a lot of experiments since, really groundbreaking experiments showing definitively that the compounds are polypropionates, that if you feed the slugs with propionate, these precursors are incorporated into the polyketide scaffold. So why that matters is when we think about animal polyketide biosynthesis or fatty acid biosynthesis, or when we think about the fungal polyketide synthesis that's known, the methyl groups usually arise or are thought to almost always arise from S-adenosylmethionine. This is a very nice example from the Fontana group where they looked at the identical compound, one produced by a fungus and the other one produced by a sacoglossan mollusk, Elysia funerea. Identical structures, the sacoglossan compound made by propionate, the fungal one made as expected by acetate labeled with methionine. So a convergent route to the same product. So to us, this was an interesting problem, something made de novo. We're not sure by who. Is it by the chloroplast, by the slug, or by some symbiotic bacteria? And with kind of bacteria-like propionate incorporation. So based upon these data, one could propose a pretty simple biogenesis coming from methylmalonyl-CoA and maybe propionyl-CoA with NADPH to give you these pyrrone-labeled polypropionates. Subsequently, methylation by an O-methyltransferase would be required. And once you have this scaffold, then you get a relatively controlled photo rearrangement to yield the natural products that are isolated from the slugs themselves. And so there are some pretty good synthetic precedents suggesting that these arise from photosynthetic rearrangements. And this literature is really interesting and recommended. In addition to being UV protective, there's literature, especially from the Moses group, describing the reaction of these compounds with reactive oxygen species, with singlet oxygen. And if you look at all of the products that have been previously isolated from the slugs, of which this is a small subset, pretty much all of their structures can be explained by a simple linear pyrrone undergoing photo rearrangements and oxidative reactions. And so both of these reactions are thought to be photoprotective and to help the slugs to maintain chloroplasts for months at a time. We're currently working on that hypothesis slowly, trying to get a handle on the right materials to test it rigorously. So far I've showed you that what was known is that these come from propionate, presumably methylmalonate, and photosynthesis. And that they're elaborated via rearrangements. And so the problem that we addressed in this project was who makes them and using what enzyme. And we really had no way of knowing in advance where this would come from, so we took a totally agnostic approach. But in the end, what we ended up with and what worked after several years of on and off trying was that we were able to use advances not done in our group, but by other groups who were doing sequencing of these organisms. And we had data obtained by others from 3 slugs, Elysia chlorotica and Elysia timida, which are long-term photosynthetic organisms and also contain these products. And a third one by Elysia cornigera, which is relatively closely related to the other 2, but it's not a long-term retainer and it does not contain these products. So we had a nice comparative system that we could dig into. We used the Elysia chlorotica transcriptome because it was the best assembled. Here are some of the references regarding the genomes and transcriptomes. When we first looked at Elysia chlorotica, we found a lot of pieces of polyketide synthases. And this is something that I think is really common in the animal genomes and transcriptomes, and that's kind of holding back the field. They're relatively hard to assemble. It's hard to find many of them that are well assembled and put together. So we obtained the raw data and kind of hand reassembled them and looked at them carefully. And using this method, complexity was brought down to 4 fatty acid synthase-like proteins. One of them was the type 2 mitochondrial Fas, which is common and which we ignored. The second one, the normal type 1 cytoplasmic Fas, which domain architecture is shown below, but which was also clearly involved in primary metabolism. And then we saw 2 Fas-like proteins that we named ECPKS1 and 2. And you can see these have very similar domain architectures, except that they're both missing a thioesterase. This experiment in the bioinformatics was nice because we could clearly map the transcripts back to the genome. So here you're seeing the schematic of the protein at the bottom, the transcript in the middle, and then mapping back to the chromosome in the background. These are still not perfect. To my knowledge, the assembly of this whole genome is still in progress. For example, we can't map half of the Fas back. We know it needs to be there because animals need the Fas to survive. But nonetheless, we could map them to the animal chromosome, showing clearly that these PKS enzymes are coming from the animals and not from the chloroplasts or from bacteria. And we did do metagenome sequencing as well, and we didn't find reasonable candidates, although you can never discount negatives in metagenomics. So looking at which fatty acid synthase might it be, you know, Adrian, especially, and a few others have looked at selectivity in bacteria. What do acyltransferases select and how do they do it? Unfortunately, in the animals and in fungi, there are no examples of methylmalonate-specific ATs, at least not that we're aware of. And therefore, we were unsure what we would find, but we were pretty lucky that one of these PKSs, ECPKS1, had a leucine valine to methionine mutation, which is correlated with preferring methylmalonate in comparison to malonate. So we thought this one seemingly might be a good candidate for expression. We wanted to make sure that we had a good candidate for expression in advance because this part is often hard. We're not the first people to try and express these types of enzymes. And then we also looked at the enoyl reductase, and both ECPKS1 and ECPKS2 looked like they would be non-functional enoyl reductases and therefore would produce polyenes. So with the guess that ECPKS1 would be important, we began investigating the other transcriptomes from Elysia timida that contains pyrones and Elysia cornigera that does not. In addition, we had 2 further species in our collection of animals that contained pyrones. And from these, we had isolated previously and purified the pyrones, and we had good analytical data showing that they were in there and which compounds were there. And so our approach was to determine whether they contained ECPKS1 homologs. Our hypothesis was that animals that contain the pyrones would express ECPKS1, whereas those that don't would not. So the answer was much cleaner than most experiments usually are. In orange are the pyrone producing long-term chloroplast retaining slugs that we looked at. In blue is the short-term one that does not contain pyrones. You can see they all contain fatty acid synthases, which is good because they all need to survive as animals. They all contain this second PKS, ECPKS2 and homologs, but only the orange ones, only the polyketide producers contained ECPKS1-like genes or transcripts. And so we felt that this was a good system for further study. So Joshua obtained this plasmid from Yi Tang. which allows expression of PKSs in Saccharomyces cerevisiae. It's got the NPGA phosphopentathionyl transferase. He synthesized and recombined this plasmid and was able to produce in yeast a little bit through some labor of the full-length 250 kilodalton ECPKS1 protein. And he was able to purify enough of that to do biochemical experiments. We also looked at whether this could produce polyketides in vivo in the yeast, and it could not. And therefore, we focused on in vitro characterization of the purified enzyme rather than in vivo work. So his analysis was to take the enzyme plus the buffer, especially methylmalonyl-CoA and NADPH. And when he did that, he was gratified to find this HPLC chromatogram in which two major compounds could be viewed, as well as a bunch of other ones. And I'll just tell you right now that compound 1 we characterized as this pyrone, which is 1 propionate unit shorter than it needs to be to make the natural products, but still is fairly compelling, I would argue. And a small amount of compound 2, which is the right chain length to be the presumed precursor for all of the sacoglossan propionates. In addition, there's a lot of this messy stuff on the baseline. What those are is rearrangement products, because these polyenes are very unstable and very difficult to handle. Because of that, we could not purify them to homogeneity and characterize them. Instead, what we had to go with was good high-res mass spectra showing that these are the accurate predicted masses of the major products. We had these 2 UV spectra for the 2 compounds, which closely correlated to our expectation. For example, here's a really nice paper from the Fontana group in which they identified, again, also using mass spec and UV because it was too unstable to purify, a somewhat similar derivative from a sacoglossan in which they proposed that this would be the precursor to many of the natural products that could be isolated. So looking similar, looking compelling, having the right features, but still unable to isolate it and characterize it further. So what we did is we decided to derivatize the enzyme reaction products. We did this using hydrogenation. And we like this method because, as you can see, you would get 4 diastereomers via this procedure. So you predict 4 diastereomers with very predictive properties. And then we could further label this for GCMS2. Excuse me. And indeed, when we ran the mass spec, gratifyingly, there were 4 isomers, and they all had the same mass. They were isobaric, and they all had the same fragmentation patterns as well. And the fragmentation patterns were not quite as good as we liked, but were well matched to the predicted structure. So we felt that we had pretty good evidence that this was the correctly identified and correctly characterized PKS. Thanks to our reviewer, we looked at this a little bit further, and we did some size exclusion chromatography showing that the big 500 kilodalton dimer is what's active, like consistent with what's known about this type of polyketides and fatty acid synthases, and that the major activity was associated with this dimer peak. And here you can even more clearly see all of these rearrangement products that we get with every assay. We investigated selectivity because, to our knowledge, although there's some data indicating that animals can take methylmalonyl-CoA in some cases, to our knowledge, there was no biochemistry showing selectivity for methylmalonyl-CoA. And so one thing that we were surprised about is there's no starter unit propionyl-CoA required. Methylmalonyl-CoA alone is what is needed. And we looked at a bunch of other conditions. And what we could see is that other CoAs actually compete with rather than improving production. So even in the case of propionyl-CoA plus methylmalonyl-CoA, the yield is not increased, but rather decreased, presumably to competition under these reaction conditions. We feel that this is good evidence that these are probably methylmalonyl-CoA selective PKSs from animals. One of the things that was really shocking to me is I always think about the great informatics that's been done by the founders of our field and continues to be done. And this informatics has led to the realization that the ketoacyl synthases, the ketosynthases from the type 1 PKSs found in animals, fungi, and bacteria are all related and share a node. In comparison here to our outgroup, the type 2 KSs, and you can see that the animal type 1 fatty acid synthases form their own branch pretty distinctively from these other enzymes, even though they share a lot of the same domain architectures. And what surprised us about this is that we expected to see if these were animal, that they would land in the animal PKS branch, but instead the sacoglossan PKSs fell into this new group that is adjacent to the animal fatty acid synthases. And I hope this is not cut off on your slide, but what we can see when we aligned more of these is that here are all of the animal polyketide synthase types represented, and bacteria and fungi. And where stars are, these are characterized at least partly PKSs. And you can see that they form a very tight cluster that is very highly supported by the data. And then up here are the animal fatty acid synthases that also form their own very tight node right here. And then adjacent to these are a bunch of enzymes that we're calling the fast type PKSs. These are mostly found so far in mollusk transcriptomes, but there are a few others like this one from a beetle. And these formed what to us looked like an unexpected new class of polyketide synthases. We think these were probably overlooked, not because they're not obvious, but because if you just